Hello and good afternoon, friends. Welcome to CEC live lectures. Dear friends, today in this session we would be discussing on one of the most interesting topics, and the topic is basics of biotechnology. Here we are going to discuss on the historical timeline of different biotechnology. So dear friends, as per the topic, we would be covering the history of biotechnology, and for this discussion we have once again with us in our studios Dr. Amit Bhattacharya. Dr. Amit Bhattacharya is Assistant Professor in Department of Zoology, Ramjas College, University of Delhi. Dear friends, if you want to ask questions from Dr. Amit Bhattacharya on basics of biotechnology, then do call us straight in the studio. You are requested to call in the last 10 minutes so that the lecture goes uninterrupted and your questions or your queries are also solved and resolved. Dear friends, you can contact us through our toll-free number. Our toll-free number is 1-800-110-430. So let's discuss on the historical perspective of the biotechnology, and let's welcome Dr. Amit Bhattacharya once again. Hello sir, welcome to the lecture.
Thank you very much, ma'am. Good afternoon, dear viewers. In this part of the lecture, I will talk about what exactly biotechnology is, how exactly we are amalgamating the different fields of biology and technology into the development of various techniques and development of various vaccines which are of very importance. Now, before going into the development of the various techniques, we should know what exactly the biotechnology is. So, basically biotechnology is an amalgamation or a combination of biological techniques as well as the technologies which are there. So, the molecular biotechnology basically covers the various fields of biology and technology, which includes molecular biology, microbiology, biochemistry, immunology, genetics, chemical engineering and cell biology. All these techniques, they combine or overlap with one another to help in the development of various transgenic crops, development of various drugs, vaccines, various different types of diagnostic kits and very hybrid kinds of livestock also. So, biotechnology is a term which is broadly used for all these technologies. Now, the major branches of biotechnologies are basically the four major branches. The first one is called as the medical biotechnology, many a times referred as the red biotechnology, which includes the improvement in the healthcare system and development of new therapies, diagnostic aids, treatment methods. The second one is the agricultural biotechnology, which is referred as the green biotechnology, which helps us to increase the yields or the crop production, helps to develop methods by which the crop protections can be done and the nutritional enrichment in the agricultural product. The third one is called as the industrial biotechnology, which is many times referred as grey or white biotechnology, which basically increases the industrial scale production using biotech tools. And the fourth one are the marine biotechnology, which is many times referred as the blue biotechnology, which uses the various biotechnological tools for the improvement of the marine and the aquatic life. Now, let's come into how exactly the biotechnology emerged out. So, the word biotechnology was coined by one of the Hungarian engineer. His name was Karl Erke. This term was coined in the year 1917. So, according to Erke, the biotechnology was all lines of work by which products are produced from raw materials with the aid of living things, unquote. Now, in the year 1961, the ambiguity ended in the year 1961 when one of the Swedish microbiologists had recommended that the title of the scientific journal dedicated for the publishing in the field of applied microbiology and industrial fermentation should be changed from Journal of Microbial and Biochemical Engineering and Technology to Biotechnology and Bioengineering. So, all these things led to the foundation of the biotechnology, and subsequently the various methods of biotechnology were developed, which helps in the production or the discovery of various diagnostic kits, various discovery of various vaccines, drugs, which which can be of importance to the medical healthcare system. Now, if we trace back the history of the biotechnology, the first timeline came in the year 1937, when one of the scientists, his name was Arne Wilhelm, he first discovered the moving boundary electrophoresis. Now, this moving boundary electrophoresis was one of the most important discoveries, which includes the complex, which helped him to isolate or differentiate between the complex nature of the serum proteins. So, the serum proteins got separated based on the charge densities. So, there were two electrodes which were put, and in the protein dissolved in the buffer, the protein dissolved in the buffer moved towards the opposite pole of the electrode. So, that is how the serum proteins were isolated, and for this breakthrough work in the year 1948, he was awarded the Nobel Prize in Chemistry. Now, these moving boundary gel electrophoresis led to the foundation of the gel electrophoresis, which is of most importance in most of the field of biotechnology, which includes the northern blotting, southern blotting, or the western blotting. Now, let us see what exactly the gel electrophoresis is. So, gel electrophoresis is basically a technique in which a mixture of DNA molecule, RNA, or the protein, it is separated based on their different sizes. So, a gel is loaded onto this gel, the various samples are loaded into each of the well, and the electrodes are then charged upon or switched on, and based on their molecular sizes they move accordingly. So, the shorter biomolecules move fastest, while the longer one remains at the top. So, that is how we can separate out the various molecules in the gel electrophoresis. Now, this is a gel electrophoresis assembly which is shown over here, and this is one of the gel electrophoresis band pattern which is shown over here, which have been kept under the UV light. So, we can see the bands which are showing the various fragments of DNA which are differing in the various sizes. Now, another breakthrough came in the year 1953, when Watson and Crick for the first time discovered or got through the breakthrough of the DNA double helix structure. So, they shared the Nobel Prize along with Wilkins for the discovery of the double helix structure or the nucleic acid. So, this was one of the most important and the most significant discovery which was made in the field of biotechnology. Now, over here we can see the first paper in which in the year 25th April 1953, the molecular structure of nucleic acid, basically the deoxyribonucleic acid, was first discovered and printed. Now, how exactly the DNA looks like? So, DNA is basically a double helix structure in which the basic backbone is formed by the ribose and the pentose. So, the basic backbone is formed by the ribose and the phosphate molecules which are present. Along with them, there are bases which are present. So, there is a base pairing which functions in the deoxyribonucleic acid base pairing rule, in which adenine binds with the thymine while the guanine binds with the cytosine. So, there is a hydrogen bonding which is found between both the bases. This hydrogen bond has been said to be the bond which is responsible for the stability of the DNA double helix structure. So, if we see the image on the left hand side of the panel, we can see Watson and Crick, with the help of their model system, they were discussing about how exactly the DNA double helix looks like and how exactly the base pairing of the DNA helix looks into. Now, another major discovery which took place in the year 1959 was the discovery of the antibodies. Now, the discovery of the antibodies was one of the most important discoveries, because antibodies were subsequently used in number of diagnostic techniques which we will come through. Now, antibodies are basically known as the immunoglobulins. So, immunoglobulins plays a very important role in the defense mechanism or the immune system in our body. So, for the discovery of the structure of the antibodies, Altman and Porter received the Nobel Prize in the year 1972 in the field of physiology or medicine. Now, basically the antibodies contains two chains. The first chain is called as the heavy chain, the other chain is called as the light chain. Each of the chain has got two areas or the regions. One is called as the variable region, while the other one is called as the constant region. So, over here we can see the variable heavy chain, while we can see in the blue color which is the constant heavy chain, while in the green color we can see the light chain, in which we can see the variable and the constant region. Now, another of the major breakthrough came in the year 1970s, when a number of scientists, they were working on the restriction endonucleases. So, restriction endonucleases are basically the enzymes which are present in the microbial microorganisms, which helps them to act as an immune system for them. So, restriction endonucleases were discovered with the help of the various techniques. So, restriction endonucleases are basically the nucleases which recognize specific sequences in the nucleic acid, and based on these recognition sequence sites, they cut the DNA segments into different fragments. Now, for this noble work, Herbert, Nathans and Smith were awarded the Nobel Prize in the year 1978. Many a times restriction endonucleases are referred as the molecular scissors in biotechnology. Now, the restriction endonucleases basically, after cutting, produces two types of ends. One is called as the sticky end, the other one is called as the blunt end. In the sticky end, there is a production of a overhanging region which is formed. So, we can see over here there is a recognition site which is GAATTC. This is the recognition site for the EcoR1 restriction endonuclease. So, EcoR restriction endonuclease cuts this sites over here, and there is a complementary overhang which is produced over there, while the other types of the restriction endonucleases basically produces the blunt ends. So, we can see over here the DNA is being cut into a blunt end. So, there is no overhangs which is present over here, and there are complete blunt ends which are produced in these types of the restriction endonucleases. Now, the restriction endonucleases have been classified into various forms or the various types. Of these, the most important one are the type 1, type 2, type 2s, type 3 and type 4. Type 1 restriction endonucleases cleaves the DNA at random sites far away from the recognition sequence, while type 2 restriction endonucleases are the most important restriction endonucleases which cleaves the DNA at the specific or designated position close or within its recognition sequences. So, type 2 restriction endonucleases are the one which are often used in the biotechnology field. Then comes the type 2s kind of restriction endonucleases which recognizes asymmetric sequences, while the type 3 cleaves outside its recognition sequences and requires two sequences in opposite orientation within the same DNA. While in the case of type 4 restriction endonuclease, it cleaves modified DNA, which is basically the methylated DNA. Now, apart from the EcoR1, which was one of the first restriction endonucleases which was discovered, there were more than 3,700 different types of restriction endonucleases which have been discovered. These different types of restriction endonucleases recognizes about different types of recognition site, which constitutes about 250 different types of recognition sites from various bacterial systems. Now, some of the restriction endonucleases sites are shown over here, in which some of the sticky ends and the blunt ends have been shown. So, the first one is a EcoR1 restriction endonucleases recognition and cleavage sequence, which is shown over here. So, we can clearly see that the EcoR1 produces a overhang and there is a sticky ends which are being produced. While in the case of HINT3 also there is a production of a overhang or a sticky end, while in the case of the HI3, which is isolated from the Haemophilus influenza, there is a production of the blunt end. So, we can see there are different cleavage pattern which is being produced by the restriction endonucleases. So, based on the sequence which we have to isolate, the biotechnologist uses the various types of the restriction endonucleases. Now, how exactly these restriction endonucleases functions? Now, these endonucleases, they, as I told you, they mostly the type 2 restriction endonucleases, they recognize specific sequences within the DNA, target DNA. These sequences are basically called as the palindromic sequences, which reads same from the back and the forth areas. So, we can see over here the recognition site is GAATTC, which is reading same from both the ends. Now, the vector DNA and the foreign DNA, both of them are cleaved with the help of the same restriction endonucleases, and as we discussed, the EcoR1 produces the restriction endonuclease sites which are sticky ends. So, both the sticky ends arms then can easily join to one another, combining or forming a recombinant form of the DNA which contains the vector DNA as well as the foreign DNA. So, this is the method by which a foreign DNA is inserted into a vector DNA part. Now, we should know why exactly the DNA of the bacterial system does not get degraded by its own restriction endonucleases. The answer is because of methylation. What the bacterial system does very intelligently is its own recognition sites, or the sites which are present within its own genome, they are methylated. Especially if we see over here, the adenine is methylated by a CH3 group. So, as soon as the adenine which is present within the recognition site of the EcoR1, it is methylated, this site cannot be recognized by the EcoR1 restriction endonucleases, and the restriction endonucleases cannot cut its own DNA. While as soon as a foreign DNA comes into or invades the bacterial system, it easily recognizes the restriction endonuclease site and it destroys or cuts the DNA into two fragments. So, the bacterial system does not destroy its own DNA because of the methylation which restricts or protects itself from the restriction enzymes functioning. Now, the restriction endonucleases, one of the most important advantage of the restriction endonucleases have been used basically in the finding out the patients who are suffering from the sickle cell anemia. Now, we know that in the sickle cell anemia there is a change or the mutation in one of the nucleotides. So, if we see the picture or the diagram over here, in the normal beta globin allele, there is a presence of two different restriction endonuclease sites which are present within the normal beta globin allele. While in the case of the sickle cell mutant beta globin allele, one of the restriction endonuclease sites is mutated, so it is no longer able to being recognized by the restriction endonucleases. So, as this restriction endonuclease is unable to recognize it, there won't be any cleavage or the cut which will be produced. So, in the case of the normal beta globin allele, if we produce a restriction endonuclease cleavage, we can find three bands which are formed over there because of the presence of two restriction endonuclease sites which are present within the normal beta globin allele. While in the case of the sickle cell mutant beta globin allele, only two band patterns are formed. So, when we run this sample onto the gel in the different wells of the gel, in the normal allele pattern we can find three different bands, while in the case of the sickle cell allele we can find only two bands. So, this clearly demarcates us that yes, the patient sample is being affected by the sickle cell anemia, while the other one is a normal allele. So, the restriction fragment analysis very critically in the initial days helped the scientists to distinguish between the normal and the sickle cell alleles of the beta globin or the hemoglobin genes. Now, another one of the breakthrough in the field of the biotechnology was the discovery of the ELISA, which is basically the enzyme linked immunosorbent assay. These discoveries were basically done in the year 1971. So, the ELISA was a technique which basically used the antibodies, and with the help of these antibodies, the various antibodies which are produced in our body, or the antigens which are present in our body, or the foreign particles which are present in our body can be identified or isolated. So, in the ELISA test, one of the most important tests which was done or developed was the HIV test. Now, what exactly is being done in the HIV test? Now, if a person is suffering from the HIV viral infection, what will happen is he or she will produce the antibodies which are very specific to the HIV antigen or the HIV virus. Now, the blood samples from the patients are collected. Now, these blood samples are then centrifuged and the serum are then loaded onto a well. These wells are pre-coated with the antigen coated wells. So, these are called as the antigen coated wells, in which the antigens are previously attached into this one. Now, the specific samples are loaded onto this well, and what will happen is the person is suffering from the HIV virus infection. There will be presence of HIV antigen specific antibodies within the sample. Now, these HIV virus antibodies will go and bind to the specific antigens which are coated into the well. Now subsequently a secondary antibody is loaded onto the well. This secondary antibody is marked with an enzyme which converts the colorless substrate into a colored product. So there is a chromogenic reaction which takes place. So as soon as there is a binding of the primary antibody to which the secondary antibody binds, we get a colored reaction. So in the well in which there is a colored reaction, we can easily identify, yes, the patient is suffering from the HIV infection, and subsequently we can titer out how much amount of the HIV antibodies which are present, or what is the viral load which is present within that particular patient. Now the second major ELISA technique was used in the identification or the detection of the malaria. Now this type of the ELISA is called as the sandwich ELISA. This ELISA basically detects the presence of the antigen, which are called as the HRP proteins or histidine rich proteins, which are produced when a patient is suffering or infected with the falciparum malaria. So these falciparum malaria antigens are present within the blood samples. Now these antigens are the blood sample from the patient is isolated. Subsequently, it is loaded onto a well which is pre-coated with the antibody which is specific to these antigen. Now suppose the patient is affected with a falciparum malaria, what will happen? There will be antigens or the proteins which will be present in his blood. Now these proteins or the antigen will come and bind to these antibodies, and subsequently a secondary antibody is loaded onto these wells. These secondary antibodies are coated with an enzyme, and this enzyme, what it does is it converts the colorless substrate into a colored product. So again a chromogenic reaction takes place, and in the wells in which we get a colored reaction, we can easily classify that yes, the patient is suffering from falciparum malaria and what exactly the disease is. So these ELISA kits has transformed the technology and the various diagnostic methods which are available for the detection of the various infections or the diseases. So these ELISA can give the results within a span of few minutes or few hours. So within a minutes or hours, we can easily identify that yes, the person is suffering from the HIV, malaria, chikungunya, dengue, or any kind of diseases for which that particular specific ELISA kit is available. Now let's talk about the emergence of the recombinant DNA technology. In the previous slides, we discovered about the main or the breakthrough techniques which were discovered in the field of biotechnology. Now the emergence of the recombinant DNA technology basically started in the year 1973 over a lunchtime conversation between the two scientists who were working in various different fields. The first scientists were Stanley Cohen, who was working on the developing different methods to transfer the plasmids, or which are basically the small circular covalently closed DNA molecules into bacterial system, while Boher in his laboratory in University of California was working on various enzyme or the restriction endonucleases that cut the DNA at specific nucleotide sequences. So over one of the conferences, they started discussing about how exactly both the techniques can be amalgamated or combined to each other for the development of recombinant DNA technology. So that laid the foundation for the emergence of the recombinant biotechnology. So over there, they discussed about what exactly are the nitty-gritties or what are the advantages, how exactly these techniques can be used. Now if we go back or trace back the history of the recombinant DNA technology, in the year 1970, Hamilton Smith at his laboratory in Johns Hopkins Medical School in USA isolated the first restriction endonucleases or the restriction enzyme, which specifically recognizes the sequence and cuts the DNA at specific nucleotide sequences. Over the several years, there were lots of different restriction endonucleases which were discovered. While in the year 1972, Cohen and Boyer combined their effort to create the recombinant DNA technology, and this recombinant DNA technology laid the foundation for the biotechnology industry. Now there were different cloning vectors which were discovered, which played a very important role in the transfer of the inserted DNA into the bacterial system. So before going into the recombinant DNA technology, we should know what exactly these cloning vectors are. One of the first cloning vector which was discovered was called as the PSC-101, which was the first cloning vector which was used in the year 1973 by Boyer and Cohen. Now, this cloning vector was of a size of 5.4 kilo base pairs and contains the antibiotic resistant tetracycline gene. There were presence of various restriction endonucleases sites within them and a origin of replication which were present. Subsequently, there were different types of the cloning vector or the plasmid vectors which were developed, which were refined form or extra forms of these cloning vectors. One of the most important discovery of the cloning vector was the PBR322. This was 4361 base pairs. It contains two antibiotic resistant genes which were present. One was ampicillin, the other one was tetracycline. And there was a presence of origin of replication. Inside the tetracycline gene, there was presence of number of restriction endonucleases sites, which were many a times referred as the MCS or multiple cloning site or polylinker sites within the plasmid DNA. So, these polylinker sites or the multiple cloning sites are the sites which are responsible for the insertion of the DNA into it. Now, let us see how exactly the recombinant DNA technology works. So, if we see the overview of the recombinant DNA technology, what we do first is we first isolate the target DNA. So, the target DNA and the plasmid cloning vectors both are restricted or digested with the help of the restriction endonucleases. As most of the restriction endonucleases produces sticky ends, what happens is they produces overhang sites. Now, with the help of the overhang sites, the target DNA and the cloning vector, they are combined to each other or ligated to each other to form the recombinant DNA combinant form. Now, subsequently, the recombinant DNA is transformed into the bacterial system where it can develop or replicate to produce the proteins of our interest. Now, before going into the fermentation process, what we require is the screening of these bacterial system, whether the recombinant DNA plasmid vector have got into that bacterial system or not. So, there are different screening techniques which are available, which includes the blue-white colony screening, colony hybridization. So, there are different techniques which are available in the market. Now, after this screening process, subsequently the bacterial cells in which the recombinant DNA have been incorporated, they are isolated. Now, these isolated form are then put into the biofermenters where they are fermented under the condition in which a number of colonies of them are produced, and subsequently these plasmid vector or the cloning vector, they replicate, transcribe and translate within the bacterial system to produce the protein of our interest. So, with the help of these recombinant DNA technology, various recombinant forms of the hormones or the proteins have been formed, which the most important one are the recombinant insulin, which generally the patients who are having high diabetes or the high sugar level are recommended. Then there is production of the recombinant growth hormone technology which is there, which is another form of the recombinant DNA technology. So, over here I conclude my lecture over here, in which I have discussed about the basic overview what exactly the recombinant DNA technology is, what are the various techniques which have been discovered in this recombinant DNA technology, and how these technologies were used subsequently for the development of the idea of recombination. So, with this note, I conclude my lecture over here. Thank you very much.
Thank you. Thank you.
dear viewers. In this second part of the lecture, which is based on basics of biotechnology, I will focus about the various biotechnology techniques which have been developed and subsequently which helped in the development of the various tools. Now, the biotechnology, the various techniques of the biotechnology includes the electrophoresis, blotting techniques, restriction and then the recombinant DNA technology, antibody structure, ELISA, DNA structure and the DNA sequences. So, a number of these techniques were discussed in the previous presentation. Now, all these techniques, they were combined to one another to develop the various techniques of biotechnology, which includes the DNA fingerprinting, polymerase chain reaction, DNA microarray, fluorescence in situ hybridization, human genome project and the human proteome projects. Now, we basically discussed about what exactly recombinant DNA technology is. So, in an overview of the recombinant DNA technology, it includes first the isolation of the target DNA, then the cut with the help of the restriction endonucleases, then the ligation or the stitching of this target DNA onto the plasmid vector, subsequently transforming these cloning vectors or the recombinant cloning vectors into the host cells and plating them onto the antibiotic containing medium, subsequently identify recombinant plasmids and characterize specific clones. Now, if we see the diagrammatic representation over here, this diagrammatic representation shows how exactly the DNA recombination techniques functions. So, there is a target DNA which is isolated or cut with the help of the restriction endonucleases and subsequently the cloning vector is also cut. Now, the plasmid cloning vector basically contains few main points. First, it contains a antibiotic resistant gene which over here is shown as the ampicillin. Now, it contains certain multiple cloning sites or the polylinker sites in which the various sites for the restriction endonucleases are present. Then the third one is the presence of the origin of replication. Now, the target DNA is then stitched or ligated onto these cloning vectors. Now, after the ligation, these cloning vectors are transformed into the bacterial host system. So, there are different transformation techniques which are available, which includes the calcium chloride or the electroporation techniques. So, with the help of these transformation techniques, what happens is these bacterial system or the host cells, they forms perforation within their cell membranes. So, as soon as the perforations are formed, these recombinant DNA forms get into the bacterial host system and they start producing the proteins from the cloned genes which are present between the bacterial host systems. Now, if we come into the history of the recombinant DNA technology into consideration, in the year 1976, Boyer co-founded a company which was the genetic technology biotechnology company, which was named as the Genentech. It was the first firm in the United States to apply for the recombinant DNA technology. And one of the first recombinant DNA technology products which was formed by this company in the year 1978 was the somatostatin. Somatostatin is basically the human growth hormone which regulates the human growth. So, somatostatin is one of the most important human growth hormone which is produced by the anterior pituitary in our body. Now, in the year 1976, Swanson and Boyer, they collaborated with one another to form a company which was Genentech with a merely funding of $500. So, that led to the foundation of the first company which subsequently became one of the most important biotechnology company of the world. Now, let us come into how exactly the recombinant form of the human growth hormone was produced. Now, somatostatin is basically a amino acid peptide which helps in the development or the cell proliferation and cell development. So, this is basically a human growth hormone and produced from the anterior pituitary. It maneuvers or inhibits the pituitary somatotropin releasing inhibitory factor. Now, these are being expressed by into or affects the various systems in our body. Now, let us see how exactly the human growth hormone was produced. Now, what the scientists found very interesting is the human gene which was for the human growth hormone, it was having a EcoR1 site present within its gene, and before that gene there was a presence of a sequence which coded for 24 amino acids, while a site which was present after this coded for 167 amino acid. Now, one of the problem with this human gene sequence coming was the presence of the signal peptide. This signal peptide are one of the most important peptide which are present in most of the genes, because the proteins which is coded from them codes with a signal peptide. Now, this signal peptide helps or acts as a carrier molecule for the transmission into the endoreticulum system. Now, what they found is whenever this whole gene was expressed or incorporated into a bacterial system, this signal peptide was also synthesized along with the human growth hormone. Now, this signal peptide was inhibiting the functioning of the human growth hormone, because this was recognized as a foreign particle as soon as they were injected within the body system. Now, what the scientists did is they very intelligently they cut the first 24 amino acid sequence gene which was present. Now, they subsequently synthesized these 24 amino acid sequence by the help of the synthetic techniques which were available, and rest of the human gene was incorporated into a plasmid vector. Into that plasmid vector, the cDNA which encoded for the 24 amino acid of the human genome growth hormone was subsequently added, and a combined or a total form of the human growth hormone was synthesized. And subsequently this was inserted into a vector, and into that vector it started expressing. And as soon as the expression started off, subsequently there was purification of these human growth hormone which took place, and they were packaged in the form of the vials for subsequent use. So, over here we can see the packaged form of the human growth hormone. Now, there were different types of the insulin which were also produced over here, and these were also produced with the help of the recombinant DNA technology. So, the next one we will discuss the importance of the recombinant DNA technology is the production of the recombinant insulin form. Now, the insulin is one of the most important hormone which is produced in our body, and this hormone regulates the glucose metabolism or the glucose concentration within the blood sample. Now, the insulin is produced by the beta cells of the islets of Langerhans from the pancreas. Now, the insulin, what it does, it inhibits the lipolysis, and subsequently it inhibits the glucose production in the case of the liver cells, but it helps in the uptake or the intake of the glucose in the muscle cells. So, what is happening is, with the production of the insulin, the blood glucose levels is maintained or lowered as soon as the increase in the blood sugar level. Now, if the insulins are not produced or they are not recognized properly, there are different characteristic forms or the disease forms which are recognized. These are called as the diabetes mellitus. In the case of a healthy person, what happens is the insulins are produced. This insulin hormone comes and binds to the insulin receptor, thereby activating the cell to uptake the glucose from the blood glucose level. Now, in the case of the type 1 diabetes mellitus, what happens is the pancreas fail to produce the insulin. So, there is no insulin production, due to which the blood glucose cannot be uptaken by the cells of the muscles or the various body parts, and subsequently the blood glucose level increases. Now, in the case of the type 2 insulin, the diabetes mellitus, what happens is the insulins are produced, but the insulin receptors get distorted and the cell fails to recognize the insulin properly with the help of its receptors. So, there is no glucose uptake and the glucose level increases. So, that is how the insulin plays a major role in our body. Now, let us see how exactly the insulins were produced. Now, insulin is basically, it contains two major polypeptide arm. One is called as the A chain, the other one is called as the B chain. So, there are two chains which are present in the insulin, and both these chains are attached to one another with the help of the disulfide bonds which are present within them. Now, what the scientists did is they isolated the human insulin gene from the pancreatic cells. Subsequently, they were ligated or cleaved with the help of the restriction endonucleases. Now, with the help of the restriction endonucleases, the insulin gene as well as the plasmid vector were cut, and subsequently they were ligated to one another with the help of the various ligases. the mixture was then formed which contains the recombinant DNA plasmids which were formed. Now, this recombinant DNA plasmid were subsequently introduced into the bacterial host system where they were developed to produce the recombinant insulin form. Now, this is a picture which is showing some of the first biofermenters of the Genentech in the year 1978, where these recombinant insulin technology was used for the production of the recombinant insulins. Now, these recombinant insulins are now marketed, and many a times they are referred as the Humulin, or they come in the packaged of bottles or the injection needles. So, these are very handy and the patient can easily put them into his or her skin to get a load of the insulin within his or her body. Now, there were development of various blotting techniques which also took place between the year 1975 to 1981, which includes the blotting technique which isolated or detected the DNA fragments, RNA fragments and the protein fragments. So, the first blotting technique which was developed in the year 1975 was the technique which helped the scientists to sequence out or isolate the DNA fragment of their interest with the help of the gel electrophoresis. This sequence technique was called as the southern blotting. Subsequently, in the year 1977, the technique which was developed for the identification and the detection of the RNA was called as the northern blotting technique, and finally the third technique was developed, which was developed in the year 1981, with the help of which the specific proteins can be isolated and they can be marked, and this technique was known as the western blotting technique. Now, if we see how exactly these techniques works, so the basic fundamental principle of all the techniques are same. So, what happens is, first we make a gel electrophoresis with the help of the gel or the agarose gel. This gel is loaded onto a cassette which is attached with a anode and a cathode. Now, onto these wells the DNA, RNA or the protein samples are loaded. Now, these electrodes are subsequently switched on. So, based upon their molecular sizes, they are separated. So, the molecular size which is smallest moves farthest, while the molecular size which is largest remains at the top. So, there is a separation in the form of the bands. Now, these gel bands are subsequently transferred onto the nitrocellulose membrane with the help of the various transferring techniques. Now, over here a cassette technique which is shown over here, in which from the gel all these bands were transferred onto the nitrocellulose membrane, and now this membrane has got the DNA, RNA or the protein of our interest. Now, let us see how exactly the southern blotting technique functions. Now, the southern blotting technique basically functions to detect the DNA of a given sequence. Now, first the DNA samples are loaded onto the gel electrophoresis assembly. Subsequently, the gel electrophoresis assembly is switched on. The gel bands are separated based on their molecular sizes. Then they are transferred onto the nitrocellulose membrane, and subsequently this membrane or the filters are loaded with the labeled DNA or the RNA probe. Now, the probe is the one, or the DNA or the RNA sequence which will specifically bind to the DNA or the RNA of our interest. So, it comes and binds to the DNA of our interest, and with the help of the autoradiography we can easily isolate the band or see the band in which the DNA of our interest is present. So, over here if we can see, there is a transcellulose image which is shown over here. In this image we can see there are few bands which are marked dark in color. These are the bands in which the probes has got attached, and they subsequently are getting lightened up. Now, the second technique which was developed was called as the northern blotting technique. This technique was basically based on the identification of the specific mRNAs. So, the mRNAs are isolated, subsequently they are loaded onto the gel, and the gel is then run. And now, the running of the gel, the various RNAs are separated, then these binding patterns are transferred onto a nitrocellulose membrane. This nitrocellulose membrane is subsequently attached or treated with the probes which are basically the RNA probes, single stranded RNA probes, and the labeled RNA are then visualized onto X-ray film, and over here if we see a X-ray film have been shown over here, in which we can see the dark bands which are showing the RNA of our interest which is present in that particular lane. Now, the third technique which was developed was called as the Western blotting technique. This technique is basically a technique which helps the scientists to identify the protein of their interest. Now, over here also what the scientists did is they first isolated all the protein samples, then the protein samples were loaded onto the gel. Now, onto this gel the electrodes were applied and the proteins were separated based on their molecular sizes. Now, based on their molecular sizes, the proteins got separated. Then from the gel, the bands were transferred onto a nitrocellulose membrane. The nitrocellulose membrane then were attached, or they were treated with the labeled antibodies. So, these labeled antibodies are basically the antibodies or the immunoglobulin which are specific to that particular protein. So, wherever the protein of our interest will be present in our band, these labeled antibodies will go and bind to that particular segment. So, as soon as we developed the autoradiography, there were X-ray images which were developed, and we can see over here the dark bands which are showing the protein of our interest, while where there is no band, it shows that the protein of our interest is not present in that particular band. Now, subsequently, there were number of techniques or the methods which were developed. Another one of the breakthrough method which was developed in the year 1977 was the development of the polymerase chain reaction. Now, along with this polymerase chain reaction, there were certain techniques which were developed which basically includes the DNA sequencing. Now, there were various scientists who were Berg, Gilbert and Sanger. They were the one who was studying the determination of the base sequences in the nucleotides. So, based on them they developed two different methods. Gilbert developed a method which is called as the sequencing method or chemical method, while Sanger along with his colleague developed a method which is called as the chain termination method. Now, both these techniques helped or made a breakthrough in the sequencing of the DNA. Now, before going into the sequencing of the DNA, we should know how exactly the DNA is being formed or it is replicated. Now, over here if we see the animated picture over here, what happens is there is a DNA strand which is there and there is a position primer which is attached. One after another the DNA nucleotide comes which is base paired with the subsequent band, and it forms a phosphodiester bond with the 3' OH of the already present ribose sugar. Now, this is how the strand is elongated. Now, this basic or very basic concept of DNA replication was used in the development of the chain termination method by the Sanger. Now, what he did is, in a normal case, in place of a base which is deoxynucleotide, what he used is a dideoxynucleotide. So, in a deoxynucleotide, the 2' OH is absent. So, there is a presence of a H, while there were certain bases which were formed which were dideoxy. So, both the second and the third carbon contained no hydroxyl group. So, if the third OH group is not present, there won't be any formation of phosphodiester bond, and the generation will be stopped or cleaved at that particular level. Now, what happened is, what the technique what he used is, first he had the DNS of which the sequence has to be elicited. So, he divided this DNA into 4 different vials. Into each of these vials the DNA template strand were added, then the DNA primer was added, DNA nucleotides were added, and to each of these wells different dideoxynucleotides were added. So, in one of the well there was an addition of DD-ATP, in one of them was the addition of DD-GTP, one of them is DD-CTP, one of them is DD-TTP. So, in the 4 tubes different dideoxy nucleotides were added. So, we know that as soon as the dideoxynucleotide will be incorporated in the place of the normal nucleotide, the chain termination will take place. So, as the chain termination took place, there were different bands or the different fragment sizes of DNA which were formed in the different tubes. all these tubes were then run into different adjacent lanes, and based on the pattern of these banding, we can elicit out the DNA sequence. So, we can see the banding pattern which is different in all the 4 tubes, and with the help of the DNA sequence, we can know what exactly the DNA sequence of our interest is. Now, one of the another interesting discoveries which was done in the year 1984 was the discovery of the DNA fingerprinting. So, DNA fingerprinting basically use the genetic approach in which the each of the organisms has got a unique pattern of microsatellites. So, each of the persons are having a unique set of repetitive sequences which are present within his gene. So, this particular technique or the idea was used by the British geneticist. His name was Alex Jeffrey. He for the first time developed the technique of DNA fingerprinting. Now, let us see what exactly DNA fingerprinting is. So, if we focus down into a small fragment or a gene fragment of a DNA, in the case of a single chromosome, we can see there are presence of different VNTRs or the variable number tandem repeats. Each of the organisms are polymorphic in the VNTR. So, each one of us are having various number of these tandem repeats present on us. So, the difference in the number of these tandem repeats make each of us unique in ourselves. So, the number of tandem repeats of a sequence is highly variable within a population. So, if we see over here between 2 restriction endonucleases cut site, in one individual there may be presence of 11 tandem repeats, in one individual there may be presence of 9 tandem repeats, while in another organisms there may be presence of 4 tandem repeats. So, based on them, each of the organisms are different. Now, this particular idea or the technique was utilized by the British scientist. So, what he did is, from the crime site, if we can get a blood sample or the sample which has got a DNA present into it. Now, this DNA was then restriction digested, and subsequently all the culprits or the person who can identified or who might have committed the crime are identified. So, the DNA samples from all these individuals are isolated. All these individuals are restricted digested with the same enzyme which is used for the DNA cut cleavage in the case of the sample which is collected from the crime site. They are run onto the gel, and the banding pattern of the person who have committed the crime and the pattern which is present from the crime site, if it matches, we can easily and confirmedly say that yes, this is the person who has committed the crime. So, if we see the image over here, we can see how the forensic sample from a site was collected, and the different 3 individuals who were suspected to have committed the crime, the DNA samples were isolated. They were restricted, digested and run onto the gel. So, if we see the banding pattern in the forensic sample site and the individual B are matching to one another. So, we can easily see that yes, the person which is the individual B has committed the crime which is of his sample was present at the forensic site. So, this was one of the major breakthrough in the biotechnology, using the biotechnological tools such as the electrophoresis, restriction endonucleases, along with the genetics which was coming into play, in which we know that all the humans population are having a variable number tandem repeats. Now, another major breakthrough which came up in the year 1987 was the discovery of the polymerase chain reaction. So, for this discovery, Mueller shared the Nobel Prize in the year 1993 in the field of chemistry. Now, polymerase chain reaction is one of the most important reaction to replicate or specifically synthesize the DNA of our interest in an in vitro condition. Now, Carey Mullis, who started working in the CTS Corporation, got the Nobel Prize in the year 1993 for his work and accredited for the invention or the discovery of the polymerase chain reaction. So, this is one of the first polymerase chain reaction which is called as a PCR 1000 thermal cycler, which was discovered and which was commercially available by the joint venture of CTS and Perkin-Elmer. nowadays there are advanced forms of the polymerase chain reaction which are available, which are called as the many times referred as the thermocyclers. So we can see how exactly the techniques developed over the years. Now again, the polymerase chain reaction use the technology of DNA replication. So in the case of the DNA replication, one after another the DNA nucleotide comes and it attaches to form a phosphodiester bond over here. Now there is a base pairing, complementary base pairing which function. So the same technique was used in the case of polymerase chain reaction, in which there were 3 different steps which are available. The first step was the denaturation, which took place at the temperature of 94 degrees Celsius, in which both these strands were separated from one another. Then the second step was the annealing, when a forward and a reverse primer were attached to these template strand, and third, when the extension started at a temperature of 72 degrees Celsius, in which the DNTPs one after another get attached based on their complementary strand. So there was a presence of a Taq polymerase also. Now if we see the animated image over here, this shows how exactly the polymerase chain reaction is functioning. So the polymerase chain reaction first starts with a denaturation step which takes place at 94, where both the strands get separated. Subsequent, there is an annealing and it takes place at a temperature of 54, and then subsequently the extension takes place at a temperature of 72 degrees Celsius. Now subsequently there were different vaccines and the development process which took place, but one of the most important techniques which helped the biotechnology field was the development of, or the discovery of the Human Genome Project. So in the year, it started in the year 1990s, and the Human Genome Project was first identified as one of the most important field. So in the Human Genome Project is basically international and collaborative research program whose main aim or the goal was mapping and understanding all the genes which are present in the human beings. So in the year 2001, the complete sequence of the Human Genome Project was published in the journal Nature, and on October 1st, 2015, it marked the 25th anniversary for the launch of the Human Genome Project. Now, in the Human Genome Project, what happened is the complete human genomes were sequenced, and these sequences helped the scientists to discover what are the changes which are taking place, what are the sites in which the changes takes place in the case of the diseases, how these diseases can be changed into a normal health condition. So the discovery or the complete sequencing of the Human Genome Project make a breakthrough in the field of sciences. So it opened a new horizon in the field of the medicines, where there were international involvement or the collaborations which took place. Subsequently, there were diagnostic testing techniques which were developed. Then the person can be given a right drug at a right place by knowing the sequence of his or her genome. Then there were various gene therapies or gene techniques which were developed, in which the healthy genes replace the distorted or the mutated gene within our body cells. So the Human Genome Project made a very huge impact in the case of the discovery or the emergence of the biotechnology. So we talked about in this part of the presentation what exactly is the importance of the human recombinant biotechnology is, how exactly this recombinant biotechnology helped in the development of the various important mechanisms like the development of the recombinant insulin form, growth hormones. Then subsequently it led to the foundation of the discovery of DNA fingerprinting, DNA microarray. Then it led to the foundation of Human Genome Project also. So with this note, I conclude my lecture. Thank you very much.
Thank you, sir. Thank you so very much for giving us a very precious session on basics of biotechnology. Dear friends, if you have any queries or suggestions regarding the lectures, specifically today's lecture, then do write to us at info.cec@nic.in. The lecture is going to be uploaded on YouTube for you very soon. Keep writing us, keep watching us. We would be meeting again soon. Till then, take care. Goodbye. Thank you, sir. Thank you once again.